Here is a table outlining the pathophysiology of demyelinating diseases through the specific autoimmune targets that contribute to each disease. First of all, neurofascin 155 and 186, which are cell adhesion molecules for structural integrity of the axon-myelin interface. They are expressed on oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system, where NF186 is localized at the nodes of Ranvier, which is implicated in nodopathies, and can be targeted through autoantibodies which disrupt node integrity, leading to demyelination and impaired conduction of action potentials. The implication of the neurofascin gene in chromosomes 1 and 13 is seen with multiple mutations and polymorphisms influencing susceptibility to demyelinating disorders such as MS, CIDP, and MMN. Contactin 1, another cell adhesion molecule involved in the formation of the paranodal junctions, is important in facilitating the organization of the nodes of Ranvier by interacting with neurofascin and Caspar-1. Autoantibodies against contactin 1 disrupt neuronal conduction and myelin, with this implication of the contactin gene on chromosome 3 with various predisposing individuals to autoimmune targeting in CIDP and other neuropathies. Gangliosides are gangliosphingolipids presented in the neural membrane, playing a role in cell signaling and membrane stability. These have four types of GM1, GD1A, GD1B, and GT1A, which are common targets in Guillain-Barré syndrome, CIDP, MMN, and with anti-GM1 antibodies predominantly associated with AMAN, a subtype of GBS, and MMN, leading to axonal damage. The implication of the GM1 synthase, an enzyme involved in ganglioside biosynthesis and polymorphisms, may affect ganglioside expression and susceptibility to autoimmune attacks. The myelin basic protein and the proteolipid protein are important myelin proteins and have been really implicated as key structural components of the myelin sheath, but also as major immune targets. And autoantibodies here lead to myelin degradation, impairing nerve signal transmission. The MBP gene on chromosome 18 has shown polymorphisms which can influence MS susceptibility and the disease course, and the PLP1 gene on chromosome X with mutations is associated with Pelizaeus-Merzbacher disease and possibly multiple sclerosis as well. Finally, with looking at the aquaporin 4 channel, we have the myelin 0 protein gene. The MPZ, myelin protein 0 chromosome on chromosome 1 is a major structural protein of the peripheral nervous system myelin sheath, involved in maintenance of the myelin sheath compactness and stability, and anti-PO antibodies contribute to PNS demyelination in conditions like CIPD and GBS. Mutations here can lead to Charcot-Marie-Tooth disease type 1b and influence autoimmune susceptibility. Also, I think it's worth to mention the PMP22 gene on chromosome 17, involved in the myelin sheath formation and maintenance as well, and is critical for its compaction, where autoantibodies here are implicated in certain PNS demyelinating disorders. And here, in Charcot-Marie disease tooth 1b, variations can influence the autoimmune targeting. Done. That was one table.
Yeah, we need the other tables as well.
We can go. They're not yet though. Looking more specifically at some of these autoimmune conditions. AMAN, which is a subset of Guillain-Barré syndrome and an acute an AIDP. Now, delving deeper into the individual conditions, firstly looking at peripheral nervous system autoimmune conditions. AMAN, which is a subset of GBS and AIDP, is primarily a nodopathy. AMAN is a paralytic disorder that presents with rapid onset of muscle weakness and absent reflexes. It's usually caused by previous infections with Campylobacter jejuni, especially strains that cause enteritis. MAN is diagnosed based on electrodiagnostic studies that show decreased compound muscular activation potentials (CMAP) amplitudes. Motor conduction blocks may appear early in MAN, but they usually disappear in later examinations. Acute motor axonal neuropathy, or AMAN, is a variant of Guillain-Barré syndrome that is caused by autoantibodies attacking the coating of motor neurons without causing inflammation. Autoantibodies mediate attack and attack specifically the axonema of motor fibers, most often at the nodal axonema. Looking more deeply into some of the susceptibility, we're going to look at the human leukocyte antigen, or HLA genes, which encodes parts of the major histocompatibility complex, which is crucial for antigen presentation and mediation of immune responses, present on chromosome 6. MHC are involved in presenting antigens after phagocytosis, where the APC communicates with the T lymphocyte in the lymph nodes to inform the adaptive immune response about infection and secrete other cytokines and molecules to activate a range of immune cells. HLA genes can be categorized into two classes: HLA A, B, and C, alpha 1, alpha 2, alpha 3, and beta 2 HLA genes can be categorized into two classes of 1 and 2. MHC1 is formed from HLA A, B, and C with 4 subunits, alpha 1, alpha 2, alpha 3, and a smaller beta 2 microglobulin subunit. And MHC2 from HLA DR, DQ, and DP with 2 alpha and 2 beta chains, each with 2 extracellular domains. Class 1 present endogenous antigens from within the cell to CD8 cytotoxic T cells, and Class 2 present exogenous antigens from outside of the cell to CD4 T helper cells. In addition, HLA genes and several non-HLA genes contribute to autoimmune susceptibility by influencing immune regulation, cytokine production, and tolerance mechanisms. More specifically, these are PTPN22, interleukin-2RA and interleukin-7RA, CTLA-4, and IRF5, each with their own subset of activity and mediating specific responses. Now, looking more specifically at neuromyelitis optica spectrum disorder, or NMOSD, which is an inflammatory and demyelinating disease primarily involving the optic nerves, brainstem, and spinal cord. Here, anti-aquaporin 4 antibodies attack aquaporin channel 4 on astrocytes, which causes astrocyte damage and subsequent demyelination. Anti-MOG antibodies define a subset of NMOSD patients targeting the myelin oligodendrocyte glycoprotein, which is involved in autoantigen behavior in the CNS and is involved in the maintenance of myelin, often with features of overlapping with ADEM and MS. NMO-IgG binding activates the complement system, resulting in inflammation and cell lysis. Infiltrating granulocytes and macrophages amplify this inflammatory response. Now looking at multiple sclerosis, which is one of the most prevalent autoimmune diseases affecting the CNS, characterized by its demyelination and subsequent neurodegeneration. Breakdown of the blood-brain barrier initiates the entry of autoreactive T cells into the CNS that recognize myelin antigens, leading to inflammation and demyelination. Common inflammation is targeted against MOG, MBP, PLP, neurofascin, and aquaporin 4. Activated microglia here will produce pro-inflammatory cytokines and reactive oxygen species, contributing to the associated tissue damage. Astrocytes also release chemokines that attract more immune cells, exacerbating the autoinflammation. Looking now at ADEM, which is a monophasic inflammatory demyelinating disease, which is often following an infection or vaccination. Direct infection of the central nervous system by a virus may activate a subset of T cells that were primed during an initial infection with the same organism. Myelin autoantigens may share antigenic determinants with an infecting pathogen through the process of molecular mimicry. Autoimmune targets are MOG and MBP in particular, contributing to the white matter damage from demyelination.